Thank you. So I'm going to talk again about the genetic diversity of Candidatus Phytoplasma palmicola in Africa in general, and Ghana and Mozambique in particular. So I'm not going to give, Dede gave a lot of information already. So just to talk about the context, in Africa, lethalioring type syndromes are associated with two phytoplasma, Candidatus Phytoplasma cocos Tanzania, described in Kenya, Tanzania, Mozambique, and Madagascar, and Candidatus Phytoplasma palmicola, described in Côte d'Ivoire, Ghana, Mozambique, Nigeria, and responsible probably of Kenkope disease in Togo and Krivi disease in Cameroon. Candidatus Phytoplasma palmicola belongs to the group 22, is transmitted by still unknown insect vectors, and in Mozambique is responsible for the destruction of more than 40,000 hectares of coconut in Nampula and Zambezia provinces. These 40,000 hectares were in 2009. And the disease has been observed in Mozambique, in the second area of production of coconut in the province of Inhambane in 2010. So the picture down represents industrial plantation in Zambezia, and the picture was taken in 2011. Next. So before the project, multi-locus sequence typing was developed to sustain epidemiological studies on Candidatus Phytoplasma palmicola. This chain is based on 8 housekeeping genes, so DNAC, GRB, LS, LPD, SEC-A, RSM-I, RPLV, and SEC-A. And it was applied to Ghanaian sample collected before 2010 on Mozambican samples from the northern province of Zambezia, Nampula, and Cabo Delgado, and on Nigerian samples. So you can see a representation of the haplotype network. So in Ghana, we observed 4 sequence types, 1 in Nigeria, and 3 in Mozambique. And you have the 2 groups, so the group B corresponding on sample from Ghana, and 1622A for Mozambique and Nigeria. So we observed 3 distinct populations corresponding on the location of sampling, but with a relatively low interpopulation diversity. So we wanted to know if the chain is able to monitor the evolution in the composition of the pathogen over time, and if it's able to detect new variants of the pathogen. Next. Then we wanted to know the origin, the sequence type responsible for the emergence of lethalioring type syndromes in Inhambane in Mozambique. So the multi-locus sequence typing was applied to sample collected during the Tropicsafe project. 102 Ghanaian DNA provided by Ndede for Phytoplasma palmicola subgroup 22B, and the 3 positive samples collected in the province of Inhambane in Mozambique, positive for Candidatus Phytoplasma palmicola 22A. So the method, by the difference with the DD diagnostic is the multi-locus sequence analysis is realized by direct PCR, so amplification of the 8 housekeeping gene by direct PCR. We control the quality and concentration for each amplicon. Each amplicon was double-strand Sanger sequenced, and with the 8 sequence of the for each sample, we concatenate it in one sequence and construct integral neighbor joining network. So the 8 housekeeping genes were obtained for 86 of the 102 Ghanaian DNA. So this is by direct PCR, and for the 3 Mozambican strain. So we concatenated for a sequence of 4,500 base pairs. And so you can see this table represents all the sequence type we got. So you have the haplotype. For each haplotype correspond on at least one mutation not detected before, and each sequence type correspond on the combination of the different haplotypes. So the ST1 to ST4 were observed previously in Ghana, ST5 to up to ST7 were sampled previously in Mozambique, and ST8 correspond on Nigeria. So the ST9 and ST10 are 2 new sequence types observed in Ghana, and ST11 to ST13 correspond on samples from Inhambane province in Mozambique. So for Ghana, you can see the color in the background correspond on the distribution of the samples observed in 2010. Well, before 2010. So you can see in the Volta region, so ST4, well, it's a little bit small, but ST4 didn't spread so much by opposition to ST3. In Volta region, the ST2 is still predominant, but you can see only 1 new sequence type in the middle, in orange, in the central region. And the bigger difference, well, the ST1 has still spread in Western region. So the new ST10, I talked about it before. And the new ST1, 9, is a new sequence type we was spread in the Western region with 7 strains, and probably resulting from the recombination of existing haplotypes, because the 2 sequence of the gene were existing previously, or conversion mutation with the one from Volta region. So you can see the network with the ST9 suggesting recombination, because it's between Volta region and ST2, and the ST10 just represented by 1 sample. Next. So the situation for Mozambican strain, the 3 positive samples gave 3 new sequence types, 11, 12, and 13. For ST11 and 12, they differ from the previous Mozambican strain with about 10 SNPs, suggesting an introduction far older than 2010 in Vilanculo. And it can explain also a difference of incidence in this province where the disease doesn't spread like in Zambezia. Next. So the surprising one is the ST13, very different of all of the other ones, with more than 52 SNPs. And this is comparable with the difference between Mozambican sequence type and Nigeria. So this suggests an independent introduction, not probably, well, sure not from the northern region. And this difference between Inhambane and northern region of Cabo Delgado and Zambezia can be explained by the control of transfer of coconut planting material in Mozambique between the 2 provinces. And this difference of composition justify maintenance of restriction. So to conclude, this study has increased our knowledge about the diversity of Candidatus Phytoplasma palmicola. It underlined the power of MLSHM for monitoring the disease, and underlined the need for continuous sampling and characterization of the pathogen. What is really missing is historical samples maybe the SC13 is very close of Cameroon or other strain we don't have. So the tools allow to follow, understand, and control the spread of the disease, and similar scheme need to be developed. So we have started to develop one for Palmae and one for Cocos Tanzania. And these data are very useful to assist the policies and the management against disease. Next, please. Thank you.
Okay.
Thank you.